pharmaceutical drug innovation crisis.
All right, great, thank you. Let me preface this talk by saying that this is probably a very different talk from every other talk here at Evolution. Hopefully you see that as a good thing at the end of this. Part of the reason that is because my colleagues and I are very interested in taking ideas about macroevolutionary methods, macroevolutionary concepts, and applying them to cultural systems. So moving them from strictly application of biological systems and exploring different questions. And the research I'm going to present here today is some of the research we have done on exploring diversification dynamics within the pharmaceutical industry. So one of the biggest debates in the pharmaceutical industry currently is whether the industry has undergone or is undergoing what is known as a productivity or innovation crisis. This has been widely debated in the literature, whether or not this crisis exists, what it is. And we felt that our macroevolutionary methods might be able to at least provide some insights into this particular question. The innovation or productivity crisis generally extends from this particular data, which is most often cited when talking about the innovation crisis. And what we see here generally is the number of new drugs approved by the FDA. So that's this line in kind of gray. We see a general decline in the new drugs approved by the FDA in roughly the last 15 or 20 years. This decline generally coincides then with a continual rise in research and development costs for new drugs. So that at the current time we have a large disparity between how much it costs to manufacture a drug and the actual number of new drugs that are approved by the FDA. Right now it's currently estimated that it's about $2.6 billion invested in order to get a drug approved. That's through all stages of the approval process. And so what we're doing here, what I'm kind of promoting in this particular talk, is a kind of macroevolutionary approach to asking some of these diversification questions. And this is, I think, more broadly expandable outside the pharmaceutical industry, and that talking about questions about productivity is, you can really use a macroevolutionary approach in talking about productivity in a number of different industries. Pharmaceutical drugs, we recently published a paper that looks at car evolution, and later today, in a poster session tonight, we look at Linux operating systems and App Store apps from the same type of approach. So one of the conceptual kind of parallels we need to draw is ideas of innovation and discontinuation, which is what we would often describe productivity with, and parallel those with ideas about origination and extinction. And by drawing these parallels, we can start to then access the suite of macroevolutionary methods used to ask questions about the processes of origination and extinction. So in this particular research, we really have two main research questions. The first is fairly straightforward. What is the rate of drug origination and extinction over the history of the drug industry? And then the second question is, what are the main factors that influence origination and extinction rates? So before I go into exactly how we answer the questions, I'll just give you some background on the data that we use in this particular research. Given that pharmaceutical drugs is a very heavily regulated industry, there's actually quite a bit of freely available information. All of our information comes from the Food and Drug Administration's approved drugs. It's an online database accessible at drugs at FDA. We specifically focus our analysis on drugs that are classified as new molecular entities. So when a drug is applied for, it's given a classification. Very few of the applications are actually considered new molecular entities. These are drugs that are considered kind of wholly different from any other drug that's currently out there. So kind of a significant innovation in a new drug. In essence, a new molecular entity that's being developed. So there are currently just about 1,263 new molecular entities. So basically all drugs out there come from these 1,263 new molecular entities. We look at about just around 1,100 drugs are used in analysis. The ones we're taking out were basically due to incomplete data in the FDA database. So once again, if we get back to these ideas of origination and extinction, what we kind of associate here is origination in this particular analysis is seen as the FDA approval date for a new drug application. So this is after all the drug is applied for, the clinical trials. This is kind of the final mark of approval given by the FDA. This is when the drug really enters the market after this approval date. And then we also look at ideas of extinction. And at least extinction in terms of this particular data and drug data, we look at only discontinued drugs, and the date of last correspondence from the FDA is listed as the discontinuation date. There's no official discontinuation date listed in the FDA database, but the standard protocol is that there's a discontinuation letter sent by the FDA to the pharmaceutical company, and so we're using the date that that letter was sent, the date of last correspondence. A majority of our drugs are still extant or are currently used in the marketplace, 85% of them, and so for those drugs we do not have an extinction date, just a time zero. So if we want to take a general look at this, we can see just some of what our data structure looks like. Drug name, we use the active ingredient here, so we don't use the brand names, as those tend to get removed after the patents end. The sponsor, so the pharmaceutical company that submitted the application. And there are two most important kind of data points here: the origination date, this is in years, and the extinction date. So we can see here that abacavir has an origination date about 17 years ago. It's still currently used in the market today, whereas Aviralex origination date of 12 years ago, and then it was discontinued fairly soon after that and taken off the market 10 years ago. We also attach therapeutic codes. This is basically identifying what what treatment the drug is typically used for, whether that be high blood pressure or oncology or whatever it might be. We can stack the kind of lifespans together here. So these are the lifespans of all 10,000 or 1,092 drugs. We can see that we do have some very long-lived drugs in the data set that originated or approved in the 40s and 50s. This is actually pre-FDA, but they still were approved. This is your penicillins, things like that. But a majority of the drugs here, essentially this top section here, come from about 1980 to the present. There's a general tendency for drugs that are originated earlier to have gone extinct or discontinued. Okay, so I think one of the main insights that we're trying to produce here is that we're trying to model drugs as stratigraphic lineages, essentially. And so we're borrowing from a kind of quantitative paleontology approach where our approval and discontinuation dates are used in the same way as dates of first and last occurrence. And so really using those two data points, then we're trying to understand the diversification dynamics within the drug industry. And as a social scientist, as someone who does a lot of cultural evolutionary research, this sort of approach is very novel, and I think provides a number of significant advantages that weren't present before. The first is that dates of origination and extinction are far easier to obtain, and I would argue much more reliable than creating long lists of traits, which had been the previous approach. Well, especially when we're talking about complex technologies such as pharmaceutical drugs, or like I mentioned before, cars. Identifying what are the key traits of that product would take, I mean, at least for something like cars, would take engineers. It would take a very long time to get a set of traits that we felt comfortable with. And that has been a large source of debate within cultural evolutionary research, has been discussion about what are the most important traits to use. We sort of circumvent that problem by looking at dates of origination and extinction. We also do not require a tree topology. So a phylogenetic approach has also been very common within cultural evolutionary research. That has been a very contentious issue as well, especially with the assumption of a branching or tree-like structure for cultural data as something that has been shown to work in some cases, like languages, but not necessarily in all cases. And so we also try to reduce that assumption of a tree-like or branching structure to the data, yet still ask questions about diversification. The program we use is called Pyrite. I won't go into too much detail about it. You can find many more details in these two papers by Daniele Silvestro and his colleagues. The main purpose of using this particular program is to then jointly estimate rates of origination and extinction from occurrence data. This is done in a fully Bayesian framework. One of the other real advantages that the Pyrite program has is that it utilizes the birth-death model, and this is advantageous for a conceptual reason in that we feel that this kind of more accurately reflects this process of innovation. Innovation is often very incremental. Very rarely you get radical innovations. And so we want to be able to include that dynamic in and that relationship between the kind of existing diversity of different technologies the existing diversity of those technologies out there and the innovations that extend from those in that kind of framework. And then finally, and I'll talk more about this point more specifically, we also want to then compare correlations between various factors that might influence diversification rates. And this is using a kind of complementary program called pyrate continuous, which I'll describe in a second. All right, so let's look at some of the results here. What do we see? Well, we see an origination rate that essentially decelerates through time here, slightly higher early on in the life history of the drugs. So what the values here on the y-axis are, a rate of event per lineage per time unit, so a rate of 0.2 is essentially saying at that particular time period, if we have 10 lineages existing, we would expect 2 origination events to have occurred in that particular time step, which in this case is 1 year. So we see a general decline in the origination rate through time. I think when 2 significant rate shifts occurring here in the late 1950s and occurring here in the mid-1990s. I think one of the clear things to see here is that we have a long, stable period of origination rate and a couple of minor decreases in origination rate, but nothing as significant as earlier in the life history of the industry. And I think this is important, right? We don't see any sort of major crisis event, right? We don't see any major decline, especially in the last 15 years, of drug origination rates. We also don't see any major increase in extinction rates. That's fairly stable throughout the life history of the drug industry as well. Okay, so now that we have this kind of general notion of the rates of origination and extinction, we can ask what factors might influence rates of origination. So I'm just looking at origination here and not looking at extinction. We can look at a number of different covariates, which is what we did with the help of some undergraduate researchers. We developed data basically looking at these 6 factors that we thought might be influential to the changes in origination rates in drugs: research and development costs, number of drug companies, mergers, which is the mergers and acquisitions, existing number of NMEs, number of prescriptions filled, number of new drug applications, Medicaid enrollees. You can see the different trajectories here. They're quite variable, so we can see some that have clear exponential growth. This is Medicaid enrollees, the number of prescriptions, or research and development costs. We see some that are much more variable as well. So using pyrate continuous, we can then establish different correlation parameters for how well each of these covariates or how they influence the origination rates. And we have only one that's statistically significant, and that's research and development costs here. You can also see that that correlation is negative, so as research and development costs are going up, origination rates are going down. This is a more visual way to look at that, where we can see the red line is the joint effects of the covariates on origination. So a joint effect of all these covariates is generally decreasing the origination rate, albeit fairly slightly by 0.02. And we can see that research and development costs have the kind of closest relationship to that particular effect. Also demonstrating that research and development costs are significant. These other 2 white lines that do also appear to have a somewhat of effect is Medicaid enrollees and the number of prescriptions filled. Although, as we can tell by the correlation parameters, they're not statistically significant. Okay, so what are some of the conclusions? What are the rates of drug origination and extinction? A declining origination rate, a fairly stable extinction rate through time. What are the main factors that influence origination rates? Research and development costs seem to be a strong influence on origination rates, which is something that has been discussed in the literature, but never kind of highlighted in such a quantitative sense before. And I think I just want to make a final point here, that what we're seeing is not an innovation crisis. So there is really no major decline in innovation or in origination. What we're seeing is an inability to control rising R&D costs. If anything, it's the lack of controlling those costs. That is the crisis that the pharmaceutical industry is facing. And one final pitch here at the end. The reason I come to these conferences and present this type of research is that I really feel strongly that macroevolutionary methods that are often more developed within the biological community can be utilized in cultural evolutionary research, and that really